We left off with, we were talking about these biofilms that bacteria or prokaryotic species. I don't want to specifically say bacteria. A lot of this is bacteria so far, but prokaryotic. So this is polysaccharides. So it's sticky, this biofilm, but it's organized sticky. So you have with this you have channels within the biofilm that allows for nutrients to get down to all of the bacteria, and also waste products to get out. So it does have a capacity with regard to its size, but there is some organization. So it isn't just it just isn't slime there, even though they sometimes call it slime. The other thing about it signals. It has chemical signals put out that actually invite other bacteria to come into the biofilm, so that allows for growth in that way. On the right, I didn't take this from your book, but another thing that and biofilms help in terms of that bacteria create a certain tolerance, particularly tolerance to antibiotics. One of the ways that the bacteria can do that is to create a biofilm that's a colony that the antibiotic will attack and penetrate the outer layer, certainly a stress layer, and then you have an environment that the bacteria actually can alter or can be altered, and then eventually at the bottom of this image you have these purple bacteria that can persist. They can be left over when the antibiotic is no longer able to penetrate, and so they will be left to multiply and carry on with regard to the bacteria. So the biofilm is also a protective barrier. Other metabolic cooperation, it's interesting, the prokaryotes can use environmental resources that they wouldn't be able to use as an individual cell. And what you have here is a cyanobacterium. Those are the photosynthetic cells. And what you have, and they're able to use light and carbon dioxide to create nutrients that are needed for their growth and development. And then you have embedded in there these heterocytes that are able to use nitrogen. So you have cells that can photosynthesize and form energy that way, but then you have these heterocytes that are nitrogen fixers. They have, in both processes, and these cells cooperate together in long chains. Both processes can carry on at the same time, even though normally something that fixes nitrogen would be rendered inactive in an oxygen environment. They're walled off so that they can exist around these photosynthetic cells and still use nitrogen fixation. And what happens then is you have both of these metabolic procedures going on, and there's cooperation between the two, and to form a area, or to form the capacity to have two different types of metabolic behavior going on. An overview or a slide about the diversity of prokaryotic cells. And one of the reasons why I'm going to keep this kind of brief in terms of the tree is because there are many things that aren't known about the lineage of certain bacterial domains, etc. And there's many changes as more and more is learned. So it is I guess you might say it's an ongoing process in terms of the tree that shows the relationship between major prokaryotes. This tree is based on molecular data. However, the more we learn about molecular data, this tree tends to be tweaked or altered based on new information. There was new information that came out in the 70s, the ability to use RNA markers. And then in the 80s, there was more development when there was the ability to use polymer chains to try to map out certain DNA sequences. And it was found that much of the tree was incorrect because of that, and so that there were some changes made in the tree and of the relationship with regard to species. They really don't know where certain species came from, where the original, they don't know what the universal ancestor is, but they're trying to get that information more close to that original ancestor, so that universal ancestor. And then the other thing is, remember we talked about transformation, transduction, and conjugation. There's been such a trading of DNA. About 75% of the DNA over the generations of bacteria has been traded. It's very difficult to trace anything back to an original ancestor. So the good news for you is probably not any questions will be asked about this tree, because we do talk about trees and lineages throughout this whole semester of biology. We will bring this back, probably bring the scheme back in different ways in different areas. Comparison of the three domains. So we do have bacteria, archaea, and eukaryotes. It's more of a review. Whether there's a nuclear envelope, and the thing about archaea, it used to be all in the bacterial family, but they found that, and again, understanding different things about it, it has some more, it seems like it's closer relative as a eukaryote than a bacteria. So no nuclear envelope in bacteria and archaea, but yes in eukaryotes. Membrane bound organelles, no in the first two, but yes in the third. In terms of the peptidoglycan cell wall, yes in bacteria, but no in archaea and no in eukaryotes. Lipid membranes, that's a similarity between archaea and eukaryotes. And then response to antibiotics, in terms of inhibited growth, histones, circular chromosome, that's a characteristic that's the same between bacteria and archaea, but not in eukaryotes. And then growth at certain temperatures. So looking at each of the groups individually, they have something that makes them very unique, but they also have some features that are similar between the two different domains on either side of archaea. Speaking of this in general, archaea, speaking in it very specifically, the archaea is also very small, but a slightly larger than bacteria, a single circular DNA chromosome, which we tend to call a plasmid. There are plasma membranes that are unique without the peptidoglycans, so without that additional layer on the outside. The outside layer of the archaea tends to be a lipid glycan. Some of them are heterotrophs, so some of them actually will eat their organic material. Some are autotrophs, some are autotroph with regard to chemicals. None are photosynthetic, which is kind of interesting, and they aren't known to cause diseases that we know of. There is a major group, we call them extremophils, not phobes. They love extremes. Extremophils. They live in extreme environments, and actually there are a few environments that they thought there was nothing capacity for anything to live, and they found that there's some archaea living there. Depending on what the extreme is that they choose to live in, there is a lovely name for it. If there's something that can live in high temperature and a low pH, so a very acidic pH, you have thermoacidophils. I probably forgot a certain portion of that. Thermoacidophils, high temperature and acidic or low pH. Methanogens, they're anaerobic. And they're really kind of interesting. They can live in an area where everything else has sucked the oxygen out of the area. So if you live in swamps where all of the oxygen has been sucked out by plants that use oxygen, these actually live there, feed on the methane gas, and will then can use methane, or they make methane as a waste product, and they create this methane gas. You have archaea that live in high salt areas, very high salt areas, so the Dead Sea, and they call them halophiles. And then there are some traits, as I said, that archaea share with eukaryotes. So the idea behind this is we have some features of archaea to have in our head, and then this concept of extremophiles. Eukaryotes do play a great role in the biosphere. They are primary decomposers, so they break down organic matter, corpses, dead vegetation, and waste product, break down organic matter and basically return it to the soil. So they're part of that energy cycle, that decomposing energy cycle. Certainly decomposers that you find in the soil, and you have billions of them in the soil, can help fix nitrogen. That nitrogen is then very, very useful for other plants to grow. They increase nitrogen, phosphorus, and potassium availability for plants. They also can decrease the availability of certain nutrients, immobilize or decrease. And so most of the time in a beneficial way, sometimes in a not so beneficial way. And then a couple of other concepts to understand before we close out this chapter. This idea of symbiosis. This is defined as a relationship between two species that live in close contact, usually one larger, one smaller, and it's a mutually beneficial for both. And so you have a Nile crocodile here, you see the picture, and then you have a Nile, I think it's a plover bird, and what the bird is doing is picking out bacteria or particles out of the crocodile's mouth that would actually cause it to probably have tooth decay, if crocodiles get tooth decay, I guess. But the relationship is positive. The bird gets something to eat, and the crocodile gets rid of something that it doesn't want. This is the idea behind symbiosis. Mutualism is a type of symbiosis that benefits both. And so at the top you have mutualism, where two positives. You think of mutualism as the bee going to the flower, getting nectar, and moving pollen from place to place. Commensalism is where you have basically one is helped while not harming the other. And what's happening between this big and little fish is the little fish is definitely helped by being around the big fish because people don't eat him, and the big fish isn't harmed by that at all. And then the third one is a parasitism, where one organism eats the other, and so one is very much benefited and the other one is harmed. And what we have here is a mosquito biting some poor person, certainly getting a meal there, and the person is getting harmed. We will pick this up on the next one.